The first week when COVID came to Singapore, we had absolutely nothing. Our diagnostic test was history and physical examination and chest x-ray. There was absolutely nothing. And with every diagnostic test that comes on, fast forward until now, we have actually sort of 3 different groups of diagnostic tests that is clinically available. We have the PCR. Next slide. We have the PCR, we have the antigen test, and we have the serology test. Roughly, we'll split the talk into thirds because of this. And actually, although it gives more information, it can cause more confusion because these are different tests testing different parts of the patient at different time point of the disease. So hopefully at the end of this talk, we can have a 3-way marriage and we can know how to interpret these in the context of clinical care. So to first, what's PCR reaction? Polymerase chain reaction, essentially it's a test where a small amount of DNA material is amplified multiple times in the thermocycler, and the target of that gene of interest is bracketed by the primers you add in. So I just put up the genome of the SARS-CoV-2 for reference, and some of these names might seem familiar to you. When you've seen some of this report, you see ORF1A, you see E, you see N, and these are all the gene names. ORF actually means open reading frame. Open reading frame is like a start of a chapter, chapter 1, open reading frame, because that region of gene codes for nothing much except for just reading. So it just goes as open reading frame 1. Of course, there are the gene below, which is a whole mouthful. So this DNA has 30,000 base pairs, right? So it is very difficult for each test to test the whole thing because then each test will run for a long time. So we only test specific segments. Sometimes in some tests you see the E gene being tested, the N gene, all right? So moving forward, then now we quickly go to the concept of cyclic threshold. Now cyclic threshold is this concept which before COVID-19 was not very well understood or even in common language. Every time a sample goes into the thermocycler, it amplifies, right? It amplifies, actually it doubles because the DNA splits, you form 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. It's a doubling. Now, if you remember, as you double, the curve goes straight up because when you go 16, 32, the gradient is very high until the machine runs out of reagent and then it plateaus off. So the cyclic threshold is basically the number of cycle it takes for the machine to detect that there is the gene of interest. So if the gene of interest is an extremely small quantity, it's a very small quantity, then the machine has to replicate many times. So the higher the number, the lower the virus level. And high CT values can be seen in a few situations, and the words are a bit small, but essentially it can be seen in early disease, very early on, or it can be seen late in the disease, like 2 weeks out, right? You can have very high CT value, okay? So now that we understand CT value is a rough correlate to the virus level, a lot of studies have sort of follow on, and then you have this study, we have to show it, it's our local study, it's made it to the big time news. This is a study of the trend of CT value over time in a patient's course, stratified by days of illness from 1 to 28. We can see the vaccinated people in the red line would have, the unvaccinated people on the red line will show a very gentle decline, while the vaccinated people in the green line will show a more sharper decline. So studies like that say that, if you're vaccinated, you tend to shed the virus shorter, and if you shed less virus. But you see, right at the start, I would draw your attention to day 1, 2, 3, 4. It's all the same. And if you look at day number 1, you see a whole bunch of people clustered around the 30, 40. So what this graph is actually trying to say, hey, in early disease, your CT can be very high, reflecting a very low virus load. Now, there's a strange concept which I'll touch on later. A CT value of 30 here can be seen in day 1, and it can be seen on day 20, but the same 30 means different things. We'll talk about that later, all right? Okay, so what are the implications of CT value? It didn't move. The implications of CT value, First of all, it is when CT value first came on, it is actually very difficult to give very specific implication. And the reason is this: CT value has never been correlated and normalized to exact viral load. We cannot tell you CT value of 20 means that's exactly this virus concentration, because that calibration has not been done. It's like, for example, I can tell you at 100 degrees, water boils. But that is called normalization in the physical world. But such collaboration between CT value and virus load has never been done. And hence, Infectious Disease Society of America can even say something like, if you take the same sample and run it in the same lab by the same people, you can have a variation of up to 3 points. So your CT of 25, you repeat it, it can come 22 or 28. And if you take the same sample and run in 10 different labs, you can have a variation as much as 14 points. So with this kind of variation, people find it hard to correlate a certain CT number with a certain virus level. However, they did say that it roughly correlates with virus level. The higher the CT value, the lower the virus level. And with that, people say, oh, then we can use this quantitative measure to demonstrate transmissibility, but not exactly. See, in the graph on the lower right hand, you see a study in college students, and actually the spreader and the non-spreader have the same CT value. Those who spread and those who doesn't spread. However, the next slide will show something different. Of course, CT value cannot be used to predict severity, because that is interplay between the virus, the host, and the immune system and the vaccine status. All right, and you see the graph on the top right. That is a graph of the possibility of growing the virus at various CT level. So if you have CT level above 30, the chance of growing a virus is very little. Okay, but yet one must caution, when you can grow a virus from somebody's sputum, it doesn't mean that he's definitely going to be highly transmissible. All right? Okay. However, you look at this slide, CT value and transmissibility. This is a study of a Danish household member, 63,000 primary case spread to 29,000 secondary case in their household. And of course, the graph clearly shows the higher the CT value, the lesser the transmission. The lower the CT value, the higher the transmission. Why is it different from the college kids? So studies like that tells us that when we look at CT value and transmissibility, we can't look at CT value alone. We must look at when the CT value was done. In this study, these are primary infection. The CT value was done at a point of primary infection early on, whereas in the college study, it was done as a screening. So a lot of people at CT 25, they have recovered, right? So we cannot make a direct correlation. And also, the closeness of the contact matters. Of course, household contact are much closer than college contact. I mean, if you don't include dormitory action and all that funny things, right? So it's a bit different from college studies. So then people say methods of collection are important. Yes. So we all know that the nasopharyngeal swab is the gold standard. It's the highest. Everything is matched to that standard. So falling below, slightly below that standard, we have nasal, salival, oropharyngeal, and now we have a combination, which is oropharyngeal nasal. That one has been done in local setting as our routine testing. So it seems like the combination of oropharyngeal and nasal is the highest, and it goes down to oropharyngeal is the lowest. Surprisingly, saliva is quite sensitive, and so then the other thing we do about to screen big population, we do pooling. And pooling is a concept that also has not been very well studied, but because of COVID and because of the large amount of testing that we have to do every day, pooling is an effective way to save testing resources. But we must also realize pooling has some inherent loss in sensitivity, because you're taking 5 people and you're diluting. So if 1 out of 5 is a positive sample, you have diluted it 5 times, because you have 4 negative samples diluting the original one. So the sensitivity is a little bit decreased. And if you think about it, 5 times seems a lot, but to a thermocycler, to CT machine, 5 times is only a CT change of 2, because it's a logarithmic curve, you see. So 5, we call it a slight drop in sensitivity. But the efficiency of pooling is also determined by the prevalence of the disease. Of course, you understand if the disease is widely prevalent, then if you pool 10 people, and every time you pool it, every pool of 10 will be positive. And then when a pool is positive, you must take out the 10 and individually test. So when the disease prevalence is very high, pooling becomes more troublesome because can you imagine a pool of 10? A pool of 10 turns positive, and then now you must check each individual one and see which of the 10 is positive. Maybe it could be 3 of them, it could be 7 of them. But so there's actually an app to determine the optimal pool size given the sensitivity of the test, the specificity of the test, and the prevalence of disease. Right? So that's all I have to say about pooling. All right? So let's. All right, and this is the most famous the slide that everybody is waiting for, variant detection. Can we use CT value to detect variant? Right now, you know, we're in the midst of an Omicron wave, right? And in this Omicron wave, the first signal that people pick up, hey, this is something new, is the S gene dropout. Because if you've heard of this S gene dropout, then so this is exactly what it means. If you were to look at the graph on the left side, you're all familiar with this S-shaped curve I showed earlier. The blue line and the green line are the ORF1 and the N protein, and the S line is flat. So on the thermocycler, this thermocycler will detect 2 genes and not detect a third one. The red line is a control. It tells you that the machine is working, so you can ignore the red line that says MS2IPC. So that's a control. So S couldn't amplify because there is a mutation at the place where the primer sits. So if the primer cannot sit, the gene cannot amplify. Right? If the gene cannot amplify, no matter how many times you run the machine, it will not show a positive signal. So on your test result, you will show something like that. I think I've anonymized this. So you see ORF, you see a number, N you see a number, and S you see nothing. So that's the S gene dropout. And why this is so important? Because if I go back 2 years, when we first had this test, we didn't have the luxury of having tests with 3 targets because we need to produce many tests. So some tests, that was an emergency. Some tests had 1 target. And then when we asked them, why do you create a test with 1 target? People say, then we thought that the virus wouldn't mutate much. There was some talk that the virus had for RNA virus, it is not as highly mutable. So they're saying that gene is not going to mutate. So they kept a 1 target test. And when that happens, when that 1 target drops out, it actually becomes a false negative test. So now all tests should have at least 2 targets. So that's in the past. So moving forward, what does it mean? It means that when you see a test result, do look at all the 3 numbers. Do look at what the targets are being tested for. And if they're testing for 3 targets and 1 target is not there, you have a variant. You have a variant, especially if you have 2 targets and 1 target is a 15 and the other target is a nil, you may possibly have a variant. Please call somebody. All right. So now maybe I've created 500 variant detection radar in the audience. All right. So now we quickly move into antigen rapid test. Is it art or is it science? Antigen rapid test came because of the pressure to decrease the speed and the turnaround of testing, and then the cost. Of course, PCR, I mean, earlier on, if you're doing single PCR for a disease that is always in the hundreds of dollars, but because of the great increase in its economic principle, then the cost of PCR has dropped now because we are doing hundreds of thousands of PCRs. But it is still more expensive than antigen. Now, antigen rapid test is a bit, well, we're all familiar with the pregnancy test. It works on a lateral flow principle. You drip the material on one end, and by capillary reaction, it just flows across the filter paper. Important difference is there is no amplification. And inherent in the difference is because PCR, there's amplification, small amounts can amplify. ART, there's no amplification. And hence, the sensitivity cannot be as good. All right? So in this, there are many papers. I just picked up one. The overall sensitivity here is quoted as 65%. And if you were to stratify it for symptomatic and non-symptomatic, the non-symptomatic people's sensitivity can go even as low as in this study, I think 40-something%. And the symptomatic people will come up to about 70-80%. And averaging up to 65%. And this study is, I chose it because this is done on nasopharyngeal swab. This is not, so the method of testing is not to be questioned here in this study. Next slide. And if you look at, this is another study. I titled this slide, Off a Cliff After 30. It's not about age, it's about CT value. So if you look at the slide on the right-hand side, you see that at various CT value, the sensitivity is pretty good at 20, 25, and when it hits 30, it starts to drop, and when it hits 35, it falls off a cliff, right? So at high CT value, ART is not so good. And if you're looking at how about the variants, this Omicron, people ask, is Omicron less sensitive? And various studies, this study has checked on 7 different ART kits, and if you look at the last column in each group, 1 to 7 are 7 different ART. Omicron seems to do the worst in terms of sensitivity. So the higher the red bar, the higher the easier it is for the ART to detect. So Omicron, it's less sensitive in all 1 to 7. And number 1 is being used quite widely in Singapore, the PanBio by Abbott, right? Okay, now we'll talk about serology. Just a quick note, serology is mainly, textbook answer, is a marker of previous infection or vaccination. That's it. We cannot use it specifically to tell how well protected a person is. We know someone vaccinated is protected, but there is no agreed correlate of protection for infection. We cannot tell 1,000 difference in antibody means what, 500 means what. We don't have that gradient. But yet we know generally more seems better. More antibody seems to have more protection. And now this is one concept which we want to touch on. Actually, we measure antibody as a surrogate of protection, not because we wanted to, because it's the only thing we can easily measure. There are other surrogates of protection like T-cell activity, avidity of B-cell, but we couldn't measure that easily, right? So the exact timing of infection we can't really tell, but we can also use it to tell vaccination or to make vaccination or booster decisions. All right. So this slide shows that for S antibody, the higher the antibody count, the higher the avidity. Avidity is how high quality it is. So there seems to be a correlate for S antibody. The higher the count, the better the quality. So it's like trying to see how handsome a person is by looking at the height. But of course, we know there's Tom Cruise, right? Short but handsome. Okay. So as a marker of vaccination, roughly you see tables like that everywhere. 2+ means vaccinated with killed vaccine. 1+ on the S means this, and then this one is really well used in the clinical sense. But I just want to make a few points. When we talk about antibody here, we refer to IgG. We don't refer to IgM, because of the graph you can see on the lower right side, IgM actually comes up at 14 days and disappears very fast. And IgG is a more enduring and more dependable marker. And it comes up at about day 2 to 3 weeks, so 14 to 28 days. By the 4th week, most people who are previously infected or vaccinated should have detectable level of IgG, all right? Okay. So on closer look, the reason why I bring this is, how about over time? What is the waning of IgG? And we always measure spike because spike wanes slower. This is the study that shows that spike wanes slower than nucleocapsid, and nucleocapsid by 24 months in this study has only 0.8% positive rate. Of course, you use a different assay, you will get a different number, but the general trend is spike tends to hold its high level rather than nucleocapsid, right? Okay, and so now if you look at various vaccines do show a different spread of antibody levels. And on the extreme right, you have Sinovac moving all the way to the champion. On the extreme left, in the green bar, you have the mRNA vaccine, extremely high level of antibodies. But once again, we cannot really directly use that to give a correlate of protection. And the last slide, going through, we talk about breakthrough infection. And if your antibody is low, what do you do? How do you know a person is infected before? How do you look at the titer? So sometimes we use a trick called the breakthrough boost. So if you have a breakthrough infection on a vaccinated person, a very low level antibody expected, for example, after Sinovac, you're expected to have anything between 2 to 3 digit number of antibody will shoot to about 4 digits. And the same phenomenon is seen in mRNA vaccine. So if a person is infected and vaccinated and yet have a very high level, much higher than what is expected of his vaccine, you can sort of make a case that this is probably a post-boost effect from infection. All right, and with that, I end my presentation. I'll leave the rest for Q&A.